Welcome back to the deep dive. So for the last few years, we've really watched foundation models, FMs, just take over, language, vision.
Absolutely.
But in biology, it feels like they've mostly focused on the transcriptome, gene expression, right? What the
Yeah.
cell's making.
Yeah, and that's super important work, don't get me wrong. But if you really want to understand what a cell is, its identity, you kind of need to go further upstream than just the products.
Okay, upstream, like the blueprints.
Exactly. You need the epigenome. Specifically something called chromatin accessibility. We often measure this with scATAC-seq.
scATAC-seq, single cell, assay for transposase-accessible chromatin using sequencing.
Right, that's the one. It basically tells you which bits of DNA are physically open and available for the cell's machinery. It's a much more fundamental look at cell state and potential than just gene expression.
Sounds like where the real decisions are made, but I'm guessing that data, it's not like reading a book, is it? What makes scATAC-seq data so tough for standard FMs?
Oh, it's a nightmare for them, honestly. It's sort of a triple threat. First, you've got just a ton of potential features, these candidate cis-regulatory elements, or cCREs, hundreds of thousands potentially.
Hundreds of thousands, okay.
Second, the data is just unbelievably sparse. Like in any one cell, only a tiny, tiny fraction of those cCREs are actually open. Your data matrix is maybe 98% zeros.
Wow, okay, 98% empty.
Pretty much.
Yeah.
And third, the little data you do have is almost binary. We usually only see counts of 0, 1, or maybe 2. So you've got this massive scale, extreme sparsity, and almost binary signal. Traditional models just, they drown.
Right, they can't find the patterns and all that. Nothingness, basically. So this crucial biological language is there, but it's hidden. Which brings us, I think, to our main topic today, EpiAgent.
Exactly.
The first foundation model built specifically for this single-cell epigenomic data using a transformer architecture. The mission, as I understand it, is to turn that sparse mess into something useful.
That's the core idea. EpiAgent tries to overcome those challenges by thinking about the cell's regulatory state, like a sentence, a cell sentence.
Okay, a cell sentence. I like that. But how do you actually build that sentence? If most of your potential words, the cCREs, are just silent, how do you translate that noisy, sparse biological reality into something structured for a transformer?
Well, the first step was building a massive foundation. They put together what they call the human scATAC corpus.
The training data.
Right. And it's huge. About 5 million cells, which translates to something like 35 billion tokens. They pulled data from 31 different tissues, 28 data sets. It's incredibly comprehensive. This is where EpiAgent learns the basic grammar of how epigenetics works.
5 million cells. That is colossal. But still, the sparsity problem, how does the cell sentence idea deal with the fact that most cCREs aren't accessible in any given cell?
That's the really clever bit, the tokenization. EpiAgent basically ignores the zeros. It only focuses on the cCREs that are actually accessible in that cell.
Okay, so you only list the open spots, but isn't the order important?
It is. And they don't just list them randomly or by their position on the chromosome. They needed a functional order. So they borrowed a technique from search engines, actually, TF-IDF.
TF-IDF, term frequency inverse document frequency. Okay, break that down. Why use that here?
Think of it like this. TF-IDF gives each accessible cCRE a score based on how unique it is. If a cCRE is open in almost every cell type they looked at in that massive corpus, it gets a low score. It's common, not very defining.
Right.
But if a cCRE is only accessible in, say, one very specific type of neuron, its TF-IDF score is high. It's distinctive. EpiAgent then ranks the accessible cCREs for that cell in descending order based on this uniqueness score.
Ah, I see. So the cell sentence starts with the most functionally important, most defining regulatory elements for that specific cell, relative to everything else in the huge training set. You're ditching the noise and ordering by significance.
Exactly. You maximize the information density right at the start of the sentence. It's not about location, it's about functional relevance.
That's smart. Okay, so this compact, functionally ordered sentence goes into the model. EpiAgent. It's big.
Yeah, about 1.4 billion parameters. And it has 3 main parts. First, there's an embedding module. This takes that sequence of cCRE indices and turns them into embeddings, basically numerical representations. It captures both the identity of the cCRE and its rank, its position in that TF-IDF ordered sentence. Kind of like word and position embeddings in NLP.
Got it. And the main engine, the part that reads the sentence.
That's the core EpiAgent transformer. It's got 18 layers of encoder blocks. And crucially, it uses a bidirectional attention mechanism.
Bidirectional, meaning it looks forwards and backwards along the sentence.
Precisely. This lets it learn the relationships between cCREs, these co-accessibility patterns, regardless of where they are in the sequence. It figures out the regulatory network logic. The whole sentence then gets compressed down into a single special output token, the CLS token embedding. That's the model's complete representation of that cell's epigenomic identity.
A single vector representing the whole cell state.
Okay.
And the third piece.
That's the signal decoder. It's actually quite simple, just a single-layer network. Its job is to take that compressed cell embedding, that CLS token, and try to reconstruct the entire accessibility signal for all cCREs, even the ones that were zero, the ones left out of the input sentence.
So it has to prove it understood the whole picture, not just the highlights you fed it.
Exactly. It forces the model to generalize.
So how does it actually learn this general grammar during pre-training before you even show it specific tasks or labels?
It learns through two unsupervised tasks run on that huge human scATAC corpus. The first is called the cell cCRE alignment task. It's basically a prediction game. The model sees the cell embedding and has to predict whether certain specific cCREs, some accessible, some not actually belong to or align with, that cell's regulatory network. It helps the model quickly learn the connections.
Makes sense. And the second task. That uses the decoder, I bet.
You got it. It's the signal reconstruction task. Here, the model must use that decoder to recreate the original binary accessibility vector for all cCREs, just from the compressed cell embedding. By constantly forcing it to unpack that dense representation back into the sparse original signal, it learns the underlying rules of how these regulatory networks are structured. It learns to write biologically plausible sentences from memory.
Okay, pre-training makes sense. It learns the language. Now, the proof is in the pudding, right? How does EpiAgent actually perform on standard scATAC-seq analyses? What did the benchmarks show?
They tested it thoroughly on 4 different validation data sets, comparing it against existing methods. We can look at the 3 big ones: unsupervised feature extraction, supervised cell annotation, and data imputation.
Let's start with unsupervised feature extraction. So taking that super high dimensional data and boiling it down to low dimensional embeddings for clustering, finding cell types. How did it stack up?
The difference was pretty stark. When fine-tuned, EpiAgent significantly outperformed the standard tools like Systopic, SPIC-Bassett, and others. On average, they saw about a 7.2% improvement in NMI that's normalized mutual information.
A measure of cluster quality, right?
Exactly. An average of 25.975% improvement in ARI, the adjusted RAND index.
Wait, hold on. Nearly 26% better ARI.
Yeah.
That's not incremental. That's a huge leap in how well it separates the cell populations accurately.
It really is. ARI is quite sensitive to getting the groupings right. And visually, you see it too. The UMAP plots show much tighter, cleaner clusters for known cell types. It separates them better and also compresses the variation within a cell type.
So higher resolution, basically.
Much higher. This clarity even let it map out complex processes like hematopoietic differentiation, blood cell development. It could accurately predict the pseudotime, the developmental stage of cells.
Okay, impressive on clustering. What about supervised cell annotation, using known cell labels to classify new data?
Again, top performer. EpiAgent beat the next best methods by an average of 11% in overall accuracy. But what was really telling was the macro F1 score.
Macro F1. Right, that one is important.
For rare cell types, it gives equal weight to every class, no matter how small. EpiAgent improved the macro F1 by over 21.5%.
21%, so it's much, much better at correctly identifying those less common but potentially critical cell populations that other methods might just gloss over or misclassify.
Precisely. Things like rare immune subsets or maybe specific progenitor cells. That's a big deal in many biological studies.
Definitely. Okay, third task, data imputation. Given how sparse the raw data is, filling in those gaps seems crucial.
Yeah, you want to reduce bias from missing signals. EpiAgent's signal decoder proved really effective here. The imputed data led to clusters that were on average 11% better in NMI and 18% better in ARI compared to using the raw, sparse data.
So filling in the blanks actually improved the downstream analysis significantly.
It did. And critically, they checked how well the imputed signals matched the real average signals for those cell types. EpiAgent got the highest correlation scores, typically over 0.8 Pearson correlation. It demonstrates the model really learned the underlying network structure to make accurate predictions about missing data.
Okay, so it nails the fundamentals, clustering, annotation, imputation with state-of-the-art results. But the really exciting stuff, I think, is when these models go beyond just describing the data and start predicting or simulating.
Exactly. That cell sentence concept unlocks some really powerful applications because you can start editing the sentence and see what happens.
Right, like predicting perturbations,
Hmm.
simulating how a cell reacts to a drug or a gene being switched off. How do they tell EpiAgent that a change is happening?
It's quite elegant, actually. They manipulate the input tokens. For a given cell sentence, they just add an extra perturbation-specific token to that main CLS token embedding before feeding it in. It's like adding an instruction. Okay, now calculate the state under this condition.
So a little tag that says add LPS stimulus or something.
Basically, yes. And they tested this with LPS-stimulated immune cells, GMNCs. The predictions were remarkably good. They got very high R-squared values when comparing EpiAgent's predicted accessibility signals after stimulation to the actual experimental data. Especially for the cCREs that changed the most, it was an almost perfect linear relationship. The model could effectively predict the cell's epigenomic response.
That's pretty cool for known stimuli. But what about things the model hasn't seen during training, like predicting the effect of knocking out a gene it's never encountered? That sounds much harder.
It is. That requires bringing in outside knowledge. For predicting the effects of unseen gene knockouts, they did something clever. They integrated a graph neural network, GNN, that uses gene ontology, GO information.
GEGO, that's that huge database of known gene functions and relationships, right?
Exactly. The GNN uses the functional pathways and connections described in GEGO to figure out what the likely impact of knocking out that unseen gene would be, and it generates the right perturbation token for EpiAgent based on that functional understanding.
Whoa, okay. So it's not just pattern matching on the ATAC-seq data anymore. It's using external biological knowledge graph, GO, to reason about the function of a gene it hasn't directly seen perturbed, and then predicting the physical chromatin changes. That feels like a step towards real biological understanding.
It really does. And this combined approach significantly outperformed a baseline model called GEARS, which mostly predicts gene expression changes. EpiAgent is predicting the change to the underlying regulatory landscape, the chromatin accessibility itself.
Very powerful. Okay, shifting gears slightly, data integration. Anyone who works with single-cell data knows about batch effects, technical noise from different experiments making it hard to combine data sets. Can EpiAgent help clean that up?
Yes, using a similar token strategy. When combining datasets, you train EpiAgent with an additional temporary batch-specific token embedding. This token learns to absorb all the technical variations, the stuff that isn't real biology.
Like a noise sponge.
Kind of, yeah. Then, once the model is trained, you just discard that batch token. And what's left are cell embeddings that are much more comparable across the original datasets. They're batch invariant.
And they showed this works.
They did. They use standard metrics for batch correction assessment, like KBET and ILIC, on a large human brain atlas. The results showed EpiAgent effectively removed the technical noise, allowing for better integration. And once you have that clean integrated reference atlas,
you can map new data onto it accurately.
Exactly. You can take query data, even from different omics types, and map it onto the reference using methods like mutual nearest neighbors to transfer labels and understand your new cells in the context of the large atlas.
Okay, that's huge for building comprehensive cell atlases. Now let's talk about what might be the most exciting application. Simulating treatments, particularly for cancer, using this in silico.
This is where the cell sentence structure really shines. Remember, the input is just that ranked list of accessible cCRE indices. So if you want to simulate the effect of, say, a drug that targets a specific regulatory element,
you just remove its index from the sentence.
You literally just delete that cCRE's index from the input sequence. You're simulating its knockout or inactivation at the most fundamental level. They demonstrated this in clear cell renal cell carcinoma, cCRCC.
Okay, cCRCC, what did they knock out in the simulation?
They focused on the promoters, the start sites of some well-known cancer-related genes like ABCC1 and VEGF-A, but they also targeted the promoter of EGLN3, a gene known to be particularly important specifically in cCRCC.
And how did they measure if the simulated knockout worked?
They created a cancerization score for each cell, basically by comparing its EpiAgent embedding to the embeddings of known normal cells and known cancer cells from their corpus. A higher score means more cancer-like.
And the results? The
simulated knockouts consistently pushed the cells towards lower cancerization scores, but the really striking finding was that knocking out the cCRCC-specific EGLN3 promoter had a much bigger effect on reducing the cancerization score compared to knocking out the more generally accessible cancer gene promoters.
Wow. So it suggests EpiAgent could potentially be used to screen, in silico, which specific epigenetic targets might be most effective for reversing a particular cancer state.
That's the potential, yes, to rapidly test hypotheses about targeting specific parts of the regulatory network.
This is really impressive. EpiAgent seems to have genuinely tackled the core challenges of scATAC-seq data, the sparsity, the scale, by reframing it as this cell sentence and applying these powerful transformer techniques.
It's a significant step forward, and they've already built on it. There are specialized versions now, like EpiAgent B for brain tissues and EpiAgent NT for non-brain tissues.
And these allow
Zero-shot annotation, which means you can take a completely new data set that the model has never seen before, feed it into the appropriate EpiAgent variant, and get highly accurate cell type labels right out of the box. No fine-tuning needed for that specific task. They're reporting accuracies over 95% for the non-brain version.
That really lowers the barrier for analyzing this complex data. So looking ahead, what's the grand vision here? Where do foundation models like EpiAgent go next in biology?
Well, the researchers have some big ideas. One is integrating the actual DNA sequence information directly into the model, maybe building towards a multi-species FM. Another is obviously bringing in other data types, transcriptomics, proteomics, to create even richer multi-omic cell sentences.
A truly holistic view of the cell.
Exactly. But the ultimate ambition mentioned is connecting models like EpiAgent with FMs from totally different fields, natural language, computer vision, maybe even robotics. The goal seems to be paving the way for genuine multimodal intelligent agents for cells.
Multimodal intelligent agents for cells. That paints quite a picture. A future where we can simulate and interact with biological systems with incredible fidelity. It really makes you think, if you had this kind of predictive power over chromatin accessibility, what previously impossible biological question could you finally start to answer? That's definitely something to ponder as we wrap up this deep dive.